Hello and good evening or good afternoon, wherever you are in the world. I am Dr. Philip McMillan, and I have been hosting these live sessions, sharing about COVID-19, various thoughts, and so on. And this week, I have the incredible pleasure of having with me Professor Anthony J. Turner. Now, for those who don't realize, the entry receptor for SARS-CoV-2, that's the virus in COVID-19, is ACE-2, and himself and his team in 2000 found this enzyme, which was a huge breakthrough and one that I think could potentially bring us a Nobel Prize. Well, let us bring with us Anthony. Anthony, how are you this evening?
Hello, Philip. Yes, I'm well, and happy to share with everybody my thoughts on ACE-2 and COVID and where we might be going in the future with it.
Excellent, excellent. Tell us a little bit about yourself. I mean, I gave you a bit of introduction there, but where was it that you came through the system?
I went to university at Cambridge and did my first degree in biochemistry and then a PhD in biochemistry there. Then spent a year in Milan doing research, and then got a position at Leeds where I've remained ever since, working my way through the system as researcher, teacher, and as always, administrator.
Yes, and
yeah.
And yes, I was just saying that one of your students, when we put out about the post, was saying what a wonderful mentor you were. So yes, you've had many students going through your labs and so on over the years.
Yeah, I think it's about 50 PhD students over my time, and quite a lot of them have stayed in the academic world or associated with it. So that's been very positive. And of course, it's always fun to be working with young people and to teach them and to learn from them as well. So it's been an enjoyable career.
Excellent. And it's through one of those PhD students that we actually have a connection. Now, most people don't realize that I am originally grew up in Jamaica. How do you have a Jamaican connection?
Well, quite by chance, my first ever PhD student at the university, an Andrew Pearson, completed his PhD and more or less immediately applied for and got a post as lecturer at the University of West Indies in Kingston. And he's been there ever since and enjoyed life there. Shortly after he went there, we established a British Council link. The British Council had funds for this type of link across the world. And this involved an exchange program. So we had technicians and junior staff from Kingston coming to Leeds, where they helped and learnt new techniques, new technologies, and then staff from Leeds would go for 3-4 weeks each year to contribute. I went several times, enjoyed visiting a beautiful country, and contributed in terms of giving teaching programs while I was there.
Excellent. Well, I won't ask you how much you enjoyed the music and so on. We won't go into that today, but that sounds like a whole special event on itself. So let's just...
Yes, maybe when we're off there.
Yes, that's right. So, okay, and Tony, I think the critical thing that I found fascinating was, what was it about the ACE-2 that made you guys start to research it all the way back in about what, 1994, 95?
Yeah, probably the mid-1990s, but there is a long background to that, which involves a direction of research which was looking at proteolytic enzymes and metabolism in the body of key peptides that control, for example, blood pressure in the cardiovascular system, in particular an enzyme known as ACE, which I guess we might call ACE-1 these days, angiotensin converting enzyme. And this is famous as it's the major target for drugs used to treat high blood pressure. The enzyme was discovered back in 1956, and in 1971, one of the most widely used antihypertensive drugs, captopril, which is a potent inhibitor of ACE, was introduced. And we began looking at some of the properties of this enzyme, particularly in terms of how it's integrated into the membrane of the cell and its metabolic physiological role. And we've been studying that for many years. And we had colleagues at Leeds who worked on insect systems, particularly Drosophila and other insects, and they knew that in their genome, they had more than one type of ACE. They didn't know much about it or what it did, but there were at least two of them. So in the 90s, we sort of reasoned that maybe, since this is such an important, ACE is such an important enzyme, maybe us humans have another form, a separate gene that's closely related that might also be important in the cardiovascular system. So that was our background thinking, and we went looking for it. And by the mid-90s, of course, we were close worldwide to obtaining the sequence of the full human genome. So this was a big help to us. We had access with colleagues in industry who helped us in terms of being able to look for fragments of the genome in libraries that existed that might resemble ACE, but were not the same as ACE. And
so I was just going to, I was going to pause you there a minute, Tony, because for many people, they just don't know or don't understand the biochemistry around it. Is there a simple way that you could put this idea across so that the general person could know what we're talking about when we say ACE and ACE-2 and angiotensin? It just sounds like a huge mush of stuff for a non-biochemistry person.
Yes, well, ACE just stands for angiotensin converting enzyme. Angiotensin is a key small molecule that acts, and you can see in this diagram here, angiotensin 1 is formed in the body and is converted by this enzyme that sits in the cell membrane by removal of 2 amino acids to something called angiotensin 2. And that is an important molecule in the body in terms of the blood pressure, regulates blood pressure, and if we want to lower blood pressure, then we need to try and block the action of Ang2, either its formation, which we do by blocking the enzyme, or the other alternative is to block the molecule, the protein in the membrane that angiotensin interacts with, the receptor. And so these enzymes that sit in the cell control metabolism and through that our cardiovascular system, our hearts. And it turned out that we thought this ACE-2 we'd found would be very similar, since ACE was very important in terms of drugs to treat medically hypertension. ACE-2 would be similar. And we got a huge surprise when we started to study what this enzyme actually did, to find that it didn't do the same or very, very similar to ACE, but it actually counterbalanced the action of ACE. So this Ang2 that raises blood pressure was converted to another molecule called Ang1-7, which you can see indicated by the arrow, and that acts through a different receptor and actually keeps our whole cardiovascular system our blood pressure regulation and our fluid handling in the body in balance. So if we get an imbalance between these two proteins, these two enzymes, ACE and ACE-2, then we get sick, essentially.
Yes.
And so we, the first surprise we had was that they didn't act similarly, they actually opposed one another. So this is how it works in science, that you have an idea and maybe it leads to something, but suddenly you're led in a completely new direction just from some observation that you make or find. And this is what happened in this case.
Now, what's interesting about this discovery, Tony, is that you guys weren't the only ones working on it and you didn't know.
This also happens in science a lot. The other group that were working on this were from a pharmaceutical company in the States, and they had obviously had similar thoughts to us and thought that they might be able to produce a potent new drug to treat heart disease from it. And they, I guess probably because at the same time they would have access to the information about the human genome, they also hit on this and we didn't know about it. They had not published anything about it at all, of course. In the scientific world, if you're an academic group, you tend to share your results, but obviously pharmaceutical group would prefer to keep them to themselves until they've developed the work further.
So did they know about your work?
Not that I know of, no. Not that I know of. I can't recall now whether we had, I don't think we published anything about it at that time. But anyway, we went on to publish. The paper appeared in a journal in mid-2000, 21 years ago now. And their paper appeared shortly afterwards in a different journal. And clearly similar results, but they'd come about it through a very different mechanism. So it wasn't that they got identical results to us. There were some differences, and we followed up, and the work developed from there. So that was 2000. We were not virologists. We knew very little about viruses.
Yeah, I was just going to ask
that this could have anything to do with viral infection. So
now, how is that? That is incredible. So you discover it in 2000, and then in 2003, we have the first SARS epidemic in Asia. What did you guys think?
Again, as so often in science, the paper about the SARS infection came out of the blue for us because this group had been looking for what it is that the SARS virus binds to get into the cell. All viruses enter the cell through binding to some receptor, like a lock and key mechanism, and that opens the cell door and lets the virus in so it can then replicate, copy itself, and then be released.
So it's this ACE-2 that it would bind to and use this to go inside the cell.
Exactly. It binds on that, and that triggers the process by which the virus gets into the cell. So without ACE-2, the virus cannot get into the cell. So this has led to ideas that therapeutically, for treatment, you might be able in some way to interfere with the interaction of the virus with ACE-2. And certainly some groups are looking at this.
And so imagine that you didn't discover this in 2000. What do you think would have happened when SARS hit?
The focus would have been much more on the SARS side of events rather than the enzyme action itself, what the protein normally did. So that it acts as a viral receptor is like a sideshow to what the protein normally does. It's being hijacked by the virus. It's nothing to do with physiologically what the the protein does. So without that, the information would have come out after 2003, but from a different background and different concept. So we would probably still be in the same position today with the knowledge base that we have now, but it would have evolved and emerged in different ways.
No, that's quite interesting, because beyond that, you guys clearly continued to research SARS-2. I mean, not SARS-2, ACE-2. How did you then come to the serum or soluble ACE-2? This is the bit that I find nobody thinks about. What made you guys think about it?
Well, yes, we did follow up from 2003. We continued working on this protein for about 10 years, but looking at how it's regulated, where it occurs, and so on. But then, of course, the SARS epidemic, which was in comparison to today's pandemic, was a relatively small epidemic. It went away for whatever reason. I don't think we really know why it disappeared. And with time, interest in ACE-2 waned. It didn't seem to be a target from which we'd get new heart disease drugs. The virus had gone away, and it actually became quite difficult to get funding to work on the protein. And of course, it only was revived really last year when the epidemic came along. And in fact, last year, I think ACE-2 was the most published on protein in the whole world. So that's quite something.
That is within 1 year, the most published on protein. So
I think there were well over 3,000 papers on it. So yes, coming back to, you're talking about a shed form. What we mean by that is that protein, or both ACE and ACE-2, sit in the cell membrane. The active part, the enzyme part, sits outside the cell, and it is cut by enzymes within the body just above the membrane surface, as your arrow is indicating, and that releases it as a soluble protein that just floats in the bloodstream then, and it can act as a soluble form of the protein. This also occurs with ACE as well, and we'd known for many years that ACE-1 if you like, does swim in the bloodstream. It's found in a shed or soluble form, and that's important in monitoring therapy for ACE inhibition. So we knew about that, and in fact we had done some quite detailed studies to try and identify what the protein was, or what the enzyme was that cuts ACE and then ACE-2 from the cell surface. So we were key in identifying that whole process.
well, let me throw it to you. I mean, I think you've been primed a little bit, but part of what I've been trying to do for the past year is highlight the point that serum ACE-2, as you say, the shed one, must be relevant in the context of this viral infection, because the evidence suggests that it binds to the viral spike protein. And what I've been saying, myself, Professor Uhal have been saying, is that when this complex gets eaten by the immune cells, they could potentially make antibodies against ACE-2 as well. Does that make any sense?
That makes perfect sense, and I think is a very viable idea for some of the severe effects that we see in COVID, how they may be brought about. Because what we haven't touched on yet is that a key factor is that, as I said earlier, ACE and ACE-2 between them counterbalance the cardiovascular system, the heart system, blood pressure regulation. So if you get any imbalance in that, then that can lead to severe effects. And so this can include, for example, the effects you see in COVID of inflammation and all the other effects you see in COVID could in large part be due to this imbalance between the enzymes in the cell and in the blood, the peptides that they act on, and this then leads to the severe effects that are seen. Now, in turn, as you say, the ACE-2 can also then be taken up, and we can, when it's fragmented, we could develop antibodies to it as an autoimmunity. So I think that is a very rational explanation for what might happen, and I think there's accumulating evidence now that that is indeed the case. Of course, it's not the only explanation by any means for why we have the severe effects in COVID, but it is a likely and probably important mechanism. So I think that needs to be followed up further, could lead to new therapeutic approaches, and it is definitely something that we need to know more of.
Yes, it's something that I'm working towards. But let's focus a little bit with regards to one of the questions that has puzzled me, and maybe as an academic you can explain this. I can't understand why it is taking us so long to figure this out. What's happening? We have so many resources across the world. Why is this being so difficult?
I think I would disagree with you there. Actually, I think what's been remarkable over the last year has been the speed with which the biology has developed, from discovering that ACE-2 is the receptor, from looking at all the effects, the development of the vaccine, all that biology, all that science, which was unknown 12, 15 months ago, has accumulated at a much more rapid pace than science normally develops. So yes, there's still a lot we have to learn, but I don't think we can say it's been slow in developing. And the other thing that's been remarkable about the science, I think, is that how openly it's been shared across the world, which 20 years ago would not have been the case to anywhere near the same extent. Researchers, in their haste to publish, are now putting their articles up online in archiving journals before they've even been peer reviewed, so reviewed by their peers, so that the world can see the results they've got, share the knowledge, and develop further. So I think it's been quite a remarkable period of science over the last 12, 15 months.
And so where do you think how much further do we have to go? So meaning that we are still at the point where we're starting the rollout of vaccines across the world. But as I pointed out with regards to the numbers, we are only at 3.4% globally. And this is a pandemic, so it doesn't matter where any one country is. The question is, where are we as a world? How much further from a realistic point of view, I guess, based on from your scientific mindset, do we have to go?
Well, it's going to be continuing for many years in order to keep this under control. I don't believe it's going to go away. And it's effectively not just the vaccine, it is to develop therapeutics. And those have to go hand in hand with the vaccine. Clearly, we have to roll out the vaccine as widely as possible and update it regularly to counter the variants of the virus that come along that try and bypass the ACE-2 lock key mechanism. But clearly, I think, we've got a long way to go, but research will need to follow up not just on the vaccines, but also, as I said, on therapeutics, There was news out, I've heard this week, about new drugs that are being tested as possible treatments, for example, anticoagulants to treat the blood clotting effects that are seen in the disease, other treatments that deal with the other side effects of the drug. There's the disease. There's a lot of new treatments that are being tested, often with pre-existing drugs, like the success with dexamethasone in treating the disease, a relatively cheap and widely available steroid. So I think we have to roll out the vaccine worldwide. That's going to be a huge task. And clearly we've already got some wars going on in terms of how that is shared. Hopefully that can be resolved and it can be distributed across the world, because all the problems we're having with being locked down, not being able to go anywhere, travel anywhere, are not going to be resolved until there's a worldwide vaccination program successful. But alongside that, we need the drugs, and there will be drugs, not only certainly pre-existing drugs may help, but there will be new drugs developed. For example, drugs that might block the interaction of ACE-2 and the virus, or in some other way interrupt that effect, or counteract the inflammatory effects, as the dexamethasone does. I know, in addition to anticoagulants, a wide range of other drugs are being tested currently. Even statins are being looked at as possible ways of treating some of the inflammatory effects. So there's a lot of work going on worldwide, but I think it's going to be hand in hand, keeping it under control with lockdown, side by side with vaccines and new drug development, or repositioning existing drugs.
An interesting question came up here, is that because the SARS-CoV-2 virus uses multiple signaling strategies, they're thinking of drugs that are acting, I guess, a multiple signaling cascade way, like something like thalidomide. Now, I know that you're from a biochemistry background, so some of these questions are not necessarily appropriate for, or not, I shouldn't say appropriate, but may not be within your area of expertise. But I don't know if you had any thoughts on this.
Yeah, I think that is a valid approach. And anything that interrupts that signaling cascade could well be important therapeutically. And there are certainly groups out there looking at this.
And so when you think from your own personal point of view and all the work that has been done in the academic world from you, do you feel as though you're missing out by not being as much in the lab and leading research systems in the same way that you were doing in the past?
In some ways, now that it's got to an exciting point, yes, but I can sit back and reflect on it and encourage my younger colleagues to develop the work in new directions. So I've spent over 40 years in direct research field, so in some ways it's good to sit and reflect and just encourage others to look at new directions. But I don't
you don't miss the hustle and bustle?
No. And certainly not the administration that goes with it. The grant writing.
Oh, yes, I can imagine the grant writing. Most of us have no idea what that is about, but I've seen from the experience of others that is quite difficult. And a lot of your research has now shifted into dementia. And you've been doing a lot of research in the latter part of your professorial work. What made the transition from ACE into dementia? How did that happen?
Well, I guess, again, chance, luck, put it how you will, but that is often the case with science, that we worked on these proteins that break down other proteins, proteolytic enzymes, and another one we had discovered in Leeds known as neprilysin, but that doesn't matter. That turned out to have an important role in Alzheimer's disease. And this again was coincidentally discovered around 2000, that this enzyme that we'd worked on for many years, and again is involved in controlling the heart and the cardiovascular system. This enzyme is in the brain, where it also is involved in metabolizing key small peptides in the brain. But it turned out one of these peptides is called amyloid beta peptide, and this is the key product that accumulates, that triggers initially the cell death that occurs in Alzheimer's disease. So it was again a chance observation that an enzyme we had worked on for many years in other areas of biology suddenly had an impact in Alzheimer's disease, and we thought, what if? And began to pursue that role and begin to look at ways in which we might be able to stimulate the removal of this amyloid. And so, yes. Coincidentally, again, as it happened, this other enzyme, ACE or ACE-1, also turns out to have a very similar role in Alzheimer's disease, that normally it helps the body, or the brain in particular, to clear away this amyloid that accumulates and clogs up the brain and causes nerve cells to die.
Yes. I'm going to show you quickly here another point. I don't know if you can see it, about drug repurposing and new therapeutics for COVID-19. And they're pointing out that a team in Denmark found that they could make peptides that look exactly like the ACE-2 receptor to potentially act as a decoy and block the binding of the SARS-CoV-2 protein.
Yes, so there have been a number of approaches like that to try and treat the disease. If in some way you could block that interaction, there are trials going on currently using what we call recombinant ACE-2, that is ACE-2, if you like, made in the laboratory, that you could administer as a decoy that the virus binds to and is disposed of. And also, of course, one of the reasons that you get these severe effects is that ACE-2 has this important role at sitting at the surface of the cell in controlling our blood pressure, etc. But when it's taken away, as it is when the virus binds and takes it inside the cell, it's then effectively inactivated. And this leads to this severe disbalance in the angiotensin peptide system that regulates the heart. And so any decoy, as suggested by that questioner, would be a valuable approach, yes.
You had mentioned that there are other viruses that use the ACE-2, but they're not actually pathogenic.
Yes, so ACE-2, both the first SARS coronavirus and SARS-CoV-2, as we call it, both use ACE-2, but there is another coronavirus that is not particularly pathogenic that also uses it, but also some related proteins sitting in the cell surface also act as receptors for other viruses, other coronaviruses. For example, the MERS virus, the Middle East Respiratory Syndrome virus, and others as well. So it is relatively common that this family of proteins seem to have been hijacked as receptors for viruses. So once once we've sorted out how to really effectively treat the COVID through ACE-2, it may lead to more effective and quick therapies when new viruses come along as future pandemics occur. occur.
Yes. So this was a point that was asked about your views on how SARS-CoV-2 evolved through the ACE-2. That's more of a virology question, and what are the advantages of using the receptor? I don't know if you have any insight into that.
I guess convenience would be the thing. It sits there, it pokes out into the outer environment, and it must, in some way, its structure must project a convenient docking point, more convenient than other proteins. And evolution takes over then of the viruses that come along. As we see, they mutate very rapidly with all the variants that we're seeing with coronavirus. And ACE-2 is a convenient target, easy to hijack, because it just sticks out there. And this is why it's being used.
Wonderful. As we're coming to the end here now, Tony, as you reflect on where we are now, and based on, I guess, your lifetime experience, what do you see over the next year or two, and what is it that you would hope for to come out of this pandemic? Is there anything good that we will see coming out of it, even if it does require some more pain?
Globally, it will lead to huge changes, and I'm sure out of that there will be many benefits about the way we do things, particularly, I hope, about the way we look at the environment and treat the environment, because it is through mistreatment that I think these viruses rapidly mutate into forms that can move between species from animal to human. So anything that changes the biology of how we interact. Politically, hopefully, it will lead to our leaders thinking of ways to be more prepared and to invest in the science that proved so effective so far, for example, in terms of vaccine development, but also in terms of making us a more global world in which we become more open and share our resources much more fairly. So those are really big outcomes, and if that happened, that would be in some ways an optimistic outcome for the pandemic, that it leads the world to better directions in the future.
Wonderful. I think that's a great way for us to close off, and thank you for whoever's made that point. It is wonderful to hear the hope for the future, and as you said about the environment, no matter what we do, we have to think about how we handle the world as humanity. So, pleasure speaking with you, Tony. I'll just ask you to hang fire. Go ahead.
I was just going to say, the last questioner who put up something seems to be much more pessimistic than me about the positive influences that will come out. But time will tell. I remain optimistic.
Time will tell. Wonderful, yeah. Wonderful. And thank you again, Tony. I'm sure we'll have the opportunity to talk again in the future. And just stay where you are for the time being while we close off. Thank you, everyone, for watching us, and join us again, hopefully shortly, as we bring more interesting, experienced scientists across the world to talk about their opinions. Have a good evening.
Thank you.